Lab 3 Determination of bacterial numbers and bacterial growth curve. The learning outcomes of this Lab 3 are to describe different methods to quantify the number of bacteria in a given sample. Second, to determine the number of viable cells in a bacterial culture by the standard plate count techniques. Third, to demonstrate skills in measuring turbidity of a culture with a spectrophotometer and relate this to the number or biomass of bacteria. Fourth, understand the growth dynamics of a bacterial culture and identify the typical phases of a bacterial growth curve. In this experiment, we're going to obtain the generation time. The generation time is the increase of bacterial population that is during the exponential or logarithmic phases. The increase of the bacterial population usually occurs in twofold, and the bacteria divide by binary fission. The diagram above show the phases during the growth of bacteria, from the lag phase to the log or exponential or growth phase, and to the stationary phase, and finally the decline phase or death phase. We have learned this during the theory class, and at the left side it shows how bacteria divide by binary fission. To calculate generation time, we can calculate it by using the formula. The generation time equals to time divided by number of generation, or you can obtain it from the graph plot as shown above. To calculate generation time by using the formula, generation time equals to the time interval divided by the number of generation. This is the several parameters that we're going to use for the calculation. T equal to time intervals. You can calculate by subtracting the T with the T naught. And then B equals the number of bacteria at the beginning of the time interval. So that means what is the number of bacteria at the point of T? And B, the number of bacteria at the end of the time interval, where at the time point of T naught, what is the number of bacteria correspond to the time. And then to solve the number of generation, we're going to use the formula log B equal to log B capital plus N log 2, because we know that bacteria divide by binary fission and it divide by twofold. Next, substitute the number of B and also capital B and divide it by log 2, that is 0.301. So therefore, n equals to 3.3 log b divided by b capital. Or it can be solve the number of generation of time g equal to t divided by 3.3 log b divided by b capital. So for an example, using the formula g equal to t divided by 3.3 log b divided by b capital, we are going to determine what is the generation time of a bacterial population that increases from 10,000 or 1 times 10 power of 4 cells to 10 million cells. That is 1 times 10 power of 7 cells in 4 hours of growth. So now we know the the time interval, or t, is equal to 240 minutes. That is 4 times 60 minutes. So we're going to substitute it as the time interval, 240 minutes, divided by 3.3 log 10 power of 7, the number of cells at the end of the logarithmic phase, and also divide by 10 power of 4, the number of bacteria at the initial state at the logarithmic phase. So from this calculation, we know that the generation time is 24 minutes. We also can get the generation time from graph plot. For example, the optical density or absorbance at 0.2, we need to double the number of the absorbance. That means it's 0.4. We're going to extrapolate the double number of the unit of absorbance, and we're going to get the time interval. Therefore, the time interval is the generation time. So in this case, the 64 minute subtract with 43 minutes is equal to 31 minutes. So that means this bacteria require 21 minutes for one generation of binary fission. So at the 0 hour minutes, only one bacterial cell, and after 21 minutes, there will be 2 bacterial cells, and after 42 minutes, there are going to be 4 bacterial cells, and it's going to increase exponentially. So we have talked about using absorbance. So what is absorbance? Absorbance is the indirect measurement of bacterial numbers by using spectrophotometry. The figures above show how a spectrophotometer works. A light source at a certain wavelength will going to be pass through the cuvette that going to be having the sample that we're going to measure the absorbance, or the sample solution, and depending on how many light being absorbed by or scattered by the bacterial cell is going to be shown in the digital display. This equipment show the spectrophotometer that we have in the lab. It's a thermoscientific, the model of Genesis 20. So when there are bacterial cells in the solution, you're going to scatter light, and we're going to usually observe the turbid or cloudy suspension in the liquid culture. The higher number of bacteria means it have more higher optical densities or absorbance, or more turbidity of the solution. Another measurement that we're going to perform in this lab is the direct measurement of bacterial numbers by using the bacterial colony counts, or also called the standard plate count. Usually we're going to perform serial dilution, either 10-fold or 2-fold dilution. In these figures, it shows 10-fold serial dilution of the original bacterial culture by serially diluted it in 1 ml to 9 ml of diluent. So this is, the sample is serial diluted, and later it being plated on the agar plate. The serial dilution of the sample that going to produce 25 to 250 colonies on the agar will be used for the calculation of the colony forming unit. As you see from this example of petri dish of agar plate, in the first plate, we'll see the bacteria is confluently grown on the agar plate. Therefore, we are unable to count the colony. We usually term this is too numerous to count. The second plate also is too numerous to count. And the third plate also too numerous to count. And the last plate, we only have 4 cells. It's not statistically useful to be using that colonies number for the calculation of CFU per mil. Usually we term that as too few to count. So after we have counted that 4th plate, we're going to use it in the calculation of CFU. The formula is CFU per mil is equal to number of colonies that you count in the 4th plate divided by the dilution factor, where in this example, the dilution factor is 10 power of negative 4. And times the volume plated. The volume plated here is 1 mil. So therefore, for example, here, if we have 32 colonies in the plate, we're going to multiply 32 times 1 times 10 power 4 divided by 1 milliliter. So that's it for the briefing for today.